Hello, my name is Christopher Gatewood Coble. The presentation I'll be giving today will be a 3D bioprinting and its potential for kidney transplants. I'll be using a real-world scenario. Jimmy's father has been waiting for a kidney on the transplant list for years. Due to organ shortages, which are a major problem worldwide, researchers have begun to explore different options like bioprinting to help provide for more people around the world. Overview. Today, I will explain what 3D bioprinting is, how the process works, its relation to cells, tissues, organs, and how three-dimensional models are important in biology. Additionally, I will cover bioink, stem cell considerations, current research, benefit limitations, and whether this technology could become a reality. What is bioprinting? 3D bioprinting is like printing a piece of paper, but biology. Not really. It's far more complex. Bioprinting is biofabrication. It uses living cells and biomaterials to replicate human tissue. Rather than using plastic like a 3D printer, bioprinters place cells within supportive materials to recreate tissues architecture. This process should mimic, with supportive technology and living cells, natural cell interactions. According to Wywerska, Krata, and colleagues, 2026, the goal of this technology is to generate multicellular structures that effectively replicate real tissue environments. How the process works. As simply as bioprinting can be explained, it is the combination of living cells and supportive material which is identified as bioink. The printer makes each layer one by one following a pattern. This will eventually build a three-dimensional structure. Other printing models utilize the same process. The biggest hurdle to cross after printing is its viability and its functionality. Long-term survival and proper cellular behavior are critical barriers to overcome. Bioprinting methods. Multiple techniques for bioprinting exist. Extrusion-based printing is the most common and mechanical or pneumatic method to deposit filaments of cell-laden hydrogel. All techniques have downsides. Clogging with higher cell densities is common among higher resolutions, while the inkjet printing deposits droplets of bioink. Laser-assisted printing is expensive and difficult to scale, but provides high precision and cell viability. Techniques like Fresh help print softer materials like collagen with solid structural fidelity. Every method carries its own trade-offs, which must have their own pros and cons weighed against them to decide which would be the better option. Why 3D matters. Two-dimensional cell cultures simply do not make the cut. They fail to accurately replicate how cells behave inside the body. In 2D systems, cells grow flat and cannot make proper cell-to-cell matrix interactions. Three-dimensional systems mimic vivo environments, ensuring that cells can behave more naturally. Organoids and organ-on-chip platforms assist with realism by incorporating multiple cell types to include mechanical or inflammatory stimuli. Wang and colleagues 2024 emphasized that 3D systems provide more predictive models rather than 2D models, especially when used for disease research and drug testing. Bioink. Bioink plays a vital role in bioprinting because it helps decide mechanical and biochemical environments that cells experience. Collagen, gelatin, hyaluronic acid, and alkalinite are natural hydrogels and are commonly used for their variability and mechanical limitations. Polyethylene glycol is a synthetic hydrogel that offers tunable mechanical properties but does not replicate biological conditions as well as its natural counterpart. Designing a bioink requires balancing printing and biological functions. In tissue engineering contexts, cell sources like stromal and regenerative cell sources are often discussed because they are a useful strategy to support tissue repair. Meslington and colleagues, 2025, found that 3D tissue engineering models prevail over 2D cultures and animal models because they lack the complexity of human tissues. Stem or stromal cell sources are crucial supporting tissue repair and organization for 3D models. Joint-related models are discussed in the paper by synovium-derived mesenchymal stromal cells, along with different approaches and as a tool for regeneration. The main idea is that using these cell sources in 3D systems assists to better reproduce cell-cell and cell matrix interaction, ensuring the model is more useful for study. Benefits plus current challenges. The strongest benefit of bioprinting is improved disease modeling. They provide more realistic platforms for studying diseases and testing drugs. They also support personalized medicine by giving models the ability to predict patient treatment responses. Additionally, this could allow researchers to rely less on animals models, which commonly fail to replicate human physiology. Examples of current research. Research focuses heavily on autoimmune disease modeling. Wywerska and colleagues, 2026, discuss immune competent 3D models that are designed to study diseases. Some examples are rheumatoid arthritis and type 1 diabetes. Discover. The primary focus is improving biomarker discovery and drug response prediction. Similarly, Wang and colleagues 2024 discuss organoid and organ-on-chip models that recreate synovial tissue. These examples reveal that bioprinting has already taken steps of improvement in preclinical research, even if full organ replacement is not yet feasible. Future prospect. Looking forward, bioprinting should advance in disease modeling, drug screening, and personalized medicine. However, reliable vascularization, long-term, not short-term functionality, and scalable modeling must be achieved to begin human viable organ printing. Based on current evidence, large-scale printing, necessary enough to support human kidney functions, like Jimmy's father, is not plausible. Advancements of bio-oriented tissues may help benefit or improve treatment strategies and accelerate therapeutic development. These are my three references I utilized and covered my information with throughout these slides. These are the two references for the images that I used for slide 3 and slide 11.